Today we're going to be looking at replication, which is an incredibly important process that takes place in all of our cells. Whenever we need to make a copy of our cells, like we do in mitosis, we need to make a copy of the DNA so that the new cell that's being made has a copy of our DNA. So when we make a copy, that's called replicate, and we're making an identical copy, so that's important. We don't want any mistakes. And this process is going to take place in the nucleus of each one of our somatic cells as they go through mitosis. This picture shows you just a very general picture of what this would look like. So first of all, you need to have your DNA split in half so that the halves of the ladder are no longer connected. And then you notice these are the new bases. So this is a new nitrogen base. So it's going to have a sugar, it's going to have a phosphate, and it's going to have an A, a G, a C, or a T on it, depending on what it needs to pair with. And they're going to come in here and rebond with these open nitrogen bases. And then lastly, we have our finished product. Now I want you to take note, because this is definitely on the test. You need to know that first of all, we're making 2 copies of DNA, so that one can go into the new cell, one can stay in the cell that it was in. And each DNA that's made is half new, so these are the new, these blue ones here, and then the other half of the DNA strand is from the original, so we would call the old strand, and one is a new strand. So in every single cell that we have, all of our DNA that we have is one old strand with one new strand of DNA when it was replicated. Now the steps in replication are pretty easy. The first thing that has to happen, like I mentioned in the picture, is that the DNA has to separate. So we have the hydrogen bonds that have to break, and we call that unzipping. Just like if you were to zip up a jacket where the teeth are connected, unzipping it, you're going to be breaking that. So you are letting the 2 halves of the ladder kind of fall open like you would in your jacket. And the parent strands, those original red ones, are going to become the template on how we determine what the new strands are. And this is where that complement strand of DNA that we practiced in the last video, if you know one half of the DNA, you should be able to come up with the complement strand. That helps us when we replicate our DNA. So step 2, we have a enzyme that comes in, and this is an important enzyme, and that is DNA polymerase. This is the name of the enzyme. And just as a reminder, enzymes are proteins, and they speed up reactions. So they're just making this possible to happen quicker. So this DNA polymerase, its job is to add those nucleotides in and also to proofread for mistakes, because there are times where maybe we have a T on one half and it's supposed to bond with an A, but maybe it bonds with a C. Well, that DNA polymerase can proofread for mistakes just like you should before you turn in your work. Step 3, we have the new nucleotides are going to bond to the parent strand. So now we have hydrogen bonds reforming. So if we look at the 2 halves of the ladder, we had this ladder here. Let's just say A, T, and G. And this is our new one that we are putting together. So we're going to have a new T coming in to pair with the A new A coming in to pair with the T, and a new C to come in to pair with that. Now this goes on for a long time. And DNA is very long, but just to give you a quick overview. And then lastly, the DNA is going to ravel back up, because it does kind of flatten out during this process more. It's not in its double helix form, so it's going to re-spiral so that it goes back to the double helix form. And then each of your daughter DNA molecules, because now we have 2 of them, each one is made from one original parent strand with one new strand. So that's what I mentioned in the picture. You need to know that a DNA molecule is half the original parent and also half the new DNA. This picture shows you the DNA polymerase. This DNA polymerase kind of goes through, and it's breaking those hydrogen bonds that are holding the original DNA here. You also have that DNA polymerase all along the process in several different places. So it is adding those nitrogen bases in So it's kind of getting this other strand, it's adding it, and it's also checking for mistakes as it goes. So what we had was an original, in this case we started with two blue strands, and in our final we end up with a blue with a red. So we do have one new strand and one old in each of our DNA molecules.